Hi, students. Today we are going to learn about mitosis. What is the goal of mitosis? Mitosis is a type of cell division in which two genetically identical daughter cells are produced. We already learned that each cell has two phases as part of its normal cell cycle: interphase and the mitotic phase. During interphase, the cell will grow and perform its regular metabolic functions. The cell will also start preparing for cell division by replicating its DNA and doubling its organelles. Thus, at the end of interphase, the cell will have two copies of genetic material and twice the amount of its cellular material. At this stage, the cell will enter into mitotic phase for division. During mitosis, the genetic material of the cell will start to divide. Mitosis has four stages called prophase, metaphase, anaphase, and telophase. The biggest challenge here is to divide and equally distribute the genetic material between the daughter cells. We don't want one daughter cell with two copies of DNA and the other one without any DNA. The cell needs to ensure that each daughter cell gets the same amount of DNA as that of the parent cell. The first stage of mitosis is called prophase. This is the preparatory phase. During prophase, the DNA becomes tightly coiled to form a structure called chromosome. Maternal and paternal copies of these chromosomes form an X shaped structure. They're called as sister chromatids. The nuclear membrane starts to disintegrate. Do you wonder why this transformation is important? This is important because DNA exists in thread like arrangement during interphase. Interphase is a metabolic phase where DNA is actively expressing its genes. Thus, the loose arrangement makes sense in the interphase. Whereas, when it comes to meiosis, that is the prophase, the goal is to equally divide the two copies of DNA. The tightly coiled arrangement will help the DNA to perfectly divide without losing any of its parts. During metaphase, rope-like structure called spindle fibers gets attached to these sister chromatids. The chromatids gets shuttled back and forth so that they get aligned at the center of the plane, an imaginary line. Why is the spindle fiber important? These rope-like structures will pull the chromatids to both sides of the cell with equal force. So each daughter cell will get one copy. During anaphase, the chromosomes move to the opposite poles of the cell. Telophase is the final stage of mitosis. This is the reverse of prophase. The nuclear membranes start forming back again. The chromosomes will start to uncoil to form the same loosened structure as we saw in the telophase. Now the cell is ready for cytoplasmic division. The telophase will be continued by cytokinesis, during which the cytoplasm will divide and two identical daughter cells will be born. Thank you for watching the lesson. Please subscribe to my channel and press the notification bell to stay tuned.